DNA, or deoxyribonucleic acid, is a molecule that contains the genetic instructions necessary for the development, functioning, growth, and reproduction of all known living organisms and many viruses. Here's an overview of how DNA works. Structure of DNA. DNA is a double-stranded molecule shaped like a twisted ladder, known as a double helix. Each strand is made up of smaller units called nucleotides, which consist of a sugar molecule, deoxyribose, a phosphate group, and one of 4 nitrogenous bases, adenine, A, thymine, T, cytosine, C, and guanine, G. The 2 strands are connected by hydrogen bonds between complementary base pairs. Adenine pairs with thymine, and cytosine pairs with guanine. Genetic information. The sequence of nucleotide bases along the DNA molecule contains the genetic information, or genetic code, that determines an organism's traits, such as its physical characteristics, behavior, and susceptibility to diseases. This genetic code is encoded in the specific sequence of base pairs along the DNA strands. Replication. DNA replication is the process by which a cell makes an identical copy of its DNA before cell division. During replication, the 2 strands of the DNA molecule unwind and separate, and each strand serves as a template for the synthesis of a new complementary strand. The enzyme DNA polymerase adds new nucleotides to the growing DNA strand following the rules of base pairing, A with T and C with G, resulting in 2 identical DNA molecules, each containing 1 original strand and 1 newly synthesized strand. Transcription. Transcription is the process by which the genetic information stored in DNA is converted into messenger RNA, mRNA, which carries the instructions for protein synthesis to the ribosomes in the cell. During transcription, the DNA double helix unwinds, and one of the DNA strands serves as a template for the synthesis of a complementary mRNA strand. RNA polymerase, an enzyme, binds to the DNA strand and catalyzes the addition of RNA nucleotides to the growing mRNA strand following the rules of base pairing, A with U and C with G. Translation. Translation is the process by which the genetic information carried by mRNA is used to synthesize proteins. mRNA molecules are translated by ribosomes, which read the mRNA codons, sequences of 3 nucleotides, and match them with the appropriate transfer RNA, tRNA molecules carrying specific amino acids. The amino acids are then linked together in a specific order dictated by the mRNA sequence, forming a polypeptide chain, which folds into a functional protein. Overall, DNA serves as the blueprint of life, encoding the instructions for the synthesis of proteins, which are the molecular building blocks and functional molecules responsible for the structure, function, and regulation of cells and organisms. DNA replication, transcription, and translation are fundamental processes that ensure the faithful transmission of genetic information and the continuity of life.